Welcome to the IWGSC webinar series. We have a great talk today. My name is Joshua Stein. I'm a project manager for the IWGSC, and I will be the moderator today. Before we get started with Agnes's talk, I'm just going to give a quick introduction to IWGSC. So we are an international consortium with membership from 3,500 people from 80 countries and 985 institutes. And among our activities is this webinar, but if you'd like to get in on it, I would welcome you to join IWGSC, and you can use the links on the bottom of this slide. I want to thank our sponsors who provide funds that help us produce this webinar and bring it to you. So those include Arbor Biosciences, CIMMYT, Florimond Desprez, INRA-E, RAGT, and Kansas Wheat. So thank you, sponsors. So a little bit about IWGSC. So IWGSC was formed about 20 years ago with the goal to sequence and provide a genome reference for hexaploid wheat. So that was accomplished back in 2018. And since that time, the objectives of IWGSC are to continue this work and to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And in order to do this, we started a project called the Wheat Genome Variation Project, and with funding from the National Science Foundation and the European Union, and collaborators at INRA-E and the University of Georgia. But the overall goal is to preserve, discover, and mobilize genetic resources by sequencing landrace genomes that encompass the worldwide genome variations of bread wheat. So we are working diligently to do platinum quality assemblies on these land races. And we are also updating Chinese Spring. So there'll be a new version of the Chinese Spring RefSeq. In addition to the genomes, we are collecting transcriptome data and using that for gene annotation. And then finally, all of these data will be packaged in a convenient form called a practical haplotype graph that will help to catalog all the genetic variations within this panel. Okay. I want to remind you there is an upcoming webinar in October 16th. So at that time, we'll be joined by Pin Yang to discuss viral resistant cereal crops. So please register to join for that. And once again, before we get started, I just want to introduce you to the webinar dashboard. So first of all, there's a chat button. So if you have any kind of technical problem or need help or need to communicate with the organizers of this webinar, you can use the chat. At the end of the talk today, there will be a question and answer session. So if a question arises that you'd like to ask our speaker, you can use the Q&A button and submit your question. And because we have limited time, we may not be able to get to all of the questions, but we will try to prioritize the questions. And we are seeking your help in the audience. If you see a question that is particularly of interest, or you were planning to ask that same question, you can go to the Q&A tab and upvote. And so we'll be able to prioritize those questions when we get to them. And if we run out of time, there's always the possibility of contacting the speaker directly. Okay, so let's get on with our webinar. Today, I'm very happy to introduce Agnes Szabo-Hever. She's a research scientist at North Dakota State University and works also at the USDA. And the title of her talk is Novel Insights on the Genetics of Susceptibility to Necrotrophic Fungal Pathogens of Durum and Bread Wheat. Okay, so thank you very much.
Thank you. I will share my screen. Hi, everyone. First, I'd like to thank for the opportunity to present my research on this webinar series. The title of my presentation is Novel Insight on the Genetics of Susceptibility to Necrotrophic Fungal Pathogens of Durum and Bread Wheat. I did this research at the USDA ARS in Fargo, North Dakota, but currently I'm working at NDSU in the Durum wheat breeding team as a research scientist. First, I'd like to give an outline about my presentation. I will talk about the wheat populations we were working on, and then the wheat pathogens, Pyrenophora, tritici-repentis, and Parastagonospora, nodorum. Then I will talk a little bit about the methods we used, and in the next two sections, I will show you the results of genetic mapping of 10-spot resistance in durum wheat and genetic mapping of septoria, nodorum, blotch resistance in durum and spring wheat. At the end of my presentation, I will talk about related studies where the phenotypic data of this research was used to validate markers. So first, I will talk about the wheat populations. Durum wheat, Triticum durum, has 2 copies of the A and B genomes, and it has 28 chromosomes. It is grown about 13.5 million hectares globally, which is about 5% of the total wheat growing area. In the US, we have durum wheat on about 2 million hectares. The main producing regions are the Middle East, North America, Southern Europe, and North Africa. We use durum wheat mainly for pasta production, make couscous, Mediterranean breads, and different semolina-based products. The idea to develop a durum panel was initiated in 2015 with the aim to identify beneficial alleles in durum wheat germplasm originating from different countries of the world and make these alleles available for breeding programs. The panel, the global durum wheat panel, consisted of more than 2,500 tetraploid wheat lines. And this included less than 1,000 Triticum turgidum species. Out of this, we selected 510 durum lines, including 332 modern accessions and 178 land races. This panel represented durum lines from 41 countries and durum lines from Simit and Icarda. The panel was first introduced to the scientific public in 2020 by Elisabetta Mazzucatelli from Filippo Bassi's team in the Frontiers in Plant Science. And now I will talk a bit about harder spring wheat. It has the A, B, and D genomes in 2 copies with 42 chromosomes. And bread wheat is grown on more than 200 million hectares globally. But out of this, only a small area has spring wheat on it. And it's mainly in the US. In the US, we have spring wheat on about 10 million hectares. As you see on the map, from the USDA National Agricultural Statistics Service. The main growing areas are North Dakota, Montana, Minnesota, South Dakota, Washington, and Idaho. We use spring wheat to make bagels, artisan hearth breads, and pizza crust. The hardest spring wheat panel that we used in our research was part of the USDA ARS National Small Grain Collection, and it consisted of 812 accessions, including cultivars, breeding lines, cultivated accessions, land races, and genetic stocks. The panel represented lines from 88 countries, and it was introduced to the scientific public in 2015 by Marco McCaffrey in a study which was about mapping stripe rust resistance in spring wheat. And now I will talk about the wheat pathogens, Pyrenophora, tritici-repentis, and Parastagonospora, nodorum. These pathogens are necrotrophic pathogens, but let's talk first about biotrophs. The biotrophic pathogen interactions follow a classical gene-for-gene model or effector-triggered immunity. So biotrophs, they use living plants for their nutrients to grow. This leads to a resistant plant. So if the host has the dominant resistance gene allele and the pathogen has the virulence gene, that will lead to a resistant plant. In other cases, other combinations, we will get a susceptible plant. So if the host has the recessive resistance gene allele or the pathogen doesn't have the virulence gene, then the plant will be susceptible. Now the necrotrophs, so they live on dead or dying plant tissue. Their plant pathogen interactions follow a toxin-based inverse gene-for-gene model or effector-triggered susceptibility. This interaction leads to a sensitive plant. So if the host has the dominant sensitivity gene and the pathogen is producing the corresponding necrotrophic effector, that will lead to a sensitive plant. If the host has the recessive susceptibility sensitivity gene, or the pathogen doesn't produce the corresponding necrotrophic effector, then we will get an insensitive plant. Pyrenophora tritici-repentis is causing the wheat disease called tan spot. The symptoms are necrotrophic lesions surrounded by chlorotic borders, and the yield loss can go up to 50%. This pathogen has the necrotrophic effector genes called PTR-tox-A, PTR-tox-B, and PTR-tox-C. The corresponding wheat sensitivity genes are TSN1 is for PTR-tox-A, PTR-tox-2 for PTR-tox-B, or TSC2 for PTR-tox-C. You may wonder why is there a difference in name, saying N and C, TSN and TSC. It's because PTR-tox-A is inducing necrosis in the wheat, so that's where it's N, and PTR-tox-B, PTR-tox-C, they are inducing chlorosis in the wheat. In my presentation, I will mention locus name called TSR7. In that case, the name is referring to resistance, so the locus is associated with tan spot resistance. In PTR, there are 8 races described, but in my presentation, I will talk about the first 5. The isolates were categorized in these races by the necrotrophic effectors they are producing. Race 1 isolate is producing PTR tox A and PTR tox C. Race 2 isolates, they produce PTR tox A. Race 3 isolates produce PTR tox C. Race 4 isolates, they don't produce any known necrotrophic effector. And race 5 isolates, they produce PTR tox B. Most of these genes has been characterized and cloned except PTR tox C. The other pathogen, Parastagonospora nodorum, is causing the wheat disease septoria nodorum blotch or SNB. The symptoms are lens-shaped or elliptical necrotic lesions with pale yellow halo. And the yield loss can go up to 30–50% here as well. Here I list the necrotrophic effector genes and the corresponding wheat sensitivity genes. If we go through this one by one, SN-tox A is recognized by 2 genes in wheat. SN-tox1 is recognized by SNN1B1 and SNN1B2. SN-tox267 is recognized by SNN2, SNN6, and SNN7. SN-tox3 is recognized by 3 genes as well. SNN4 is recognizing SN-tox4, and SN-tox5B1 and SNN5B2 is recognizing SN-tox5. And most of these genes were characterized and cloned as well, except SN-tox4, TSN1B2, SNN4, SNN5B2, and SNN6 and SNN7. And now I will talk just a little bit about the methods we used. The global Durum panel was tested for 10-spot resistance and SNB resistance. The 10-spot resistance included infiltrations and spray inoculations. We infiltrated the Durum panel with PTR tox A and PTR tox B, and we inoculated the Durum panel with isolates representing race 1, 2, 3, 4, and 5. During the SNB resistance test, we infiltrated the Durum panel with 5 necrotrophic effectors, SN-tox5, SN-tox1, SN-tox267, SN-tox3, and SN-tox5. The same necrotrophic effectors were used to test SNB resistance in the hardest wheat panel too. And here's a picture which is showing the infiltration process. We use a needless syringe to press the culture filtrate into the leaf tissue, and we use marker to mark how far the culture filtrate went in the leaf tissue. Both panels were genotyped with the Illumina 90K SNP array. And we used R for GWAS and the MLM method in R. And now I will show you the results of genetic mapping of 10-spot resistance in durum wheat. As you see on this histogram, most of the durum lines were sensitive or moderately sensitive to the first 3 isolates. And about 1/3 of the Durum panel was sensitive, and about half of the Durum panel was moderately sensitive to the race 5 isolate DW5. And as we expected, most of the Durum lines were resistant to the race 4 isolate L13-192. And it was expected because this, as I said, this race is not producing any non-necrotrophic effector. But in our study, we were able to identify some susceptible or moderately susceptible lines, durum lines. I will not show you the geographic distribution of susceptible lines for each isolate, but here's a map that shows the average disease scores of the 5 races. And here on the map, you can see that most of the susceptible lines were from South Asia, North America, Middle East, and the Balkans. Here's a Manhattan plot we got with the race 1 isolate, PTI2, which is producing PTR tox A and PTR tox C. You can see that the TSC1 PTR tox C interaction was significant, but we didn't detect the TSN1 locus that would recognize PTR tox A. And that was expected because it was demonstrated earlier studies actually that the TSN1 PTR tox A interaction is not playing a significant role in Durum. And this was proved with our race 2 isolate. TSN1 locus wasn't significant here either. This is another PTR tox A producing isolate. But there was a locus on chromosome 3b that was significant, and that was the locus where TSR7 was mapped earlier in 2020 by Justin Ferris in wild emmer wheat. And it was shown earlier studies that this QTL or this locus is a race non-specific QTL, so it's providing resistance across races. And this is what we see with race 3. A TSR7 showed up here too. And you may wonder why we didn't get the same locus in PTI2. But it was probably, it is there, it just didn't reach the significance level. Another study that was published in 2020 on a Durum population from the National Small Grain Collection, for example, they were able to detect this locus at PTI2 with PTI2 isolate as well. And another interesting thing here with race 3 isolate that the TSC1 locus didn't reach the significance threshold. And that was interesting because you would think that because of the PTR tox C and the isolate, that would be significant. With the race 4 isolate, we found the locus significant on the short arm of chromosome 2A. This race doesn't, again, it doesn't produce any known effector. But we are not sure if it's a locus for a new sensitivity gene, or it's a locus which provides resistance like the TSR7. But it was interesting to see that the same locus was close to the significance threshold here with race 2 isolate. It just didn't reach the significance threshold. So there is a chance that this is another TSR locus. With DW5 isolate, as we expected, TSC2 locus was significant. It's a PTR tox B producer isolate. And then interestingly, a new QTL was found on the long arm of chromosome 5b. I will come back to this Manhattan plot in a few minutes, and we'll talk about this QTL a bit more. I will show you the infiltration results on the global Durum panel. You can see that with the PTR tox A, about a third of the population, 28% of the population was moderately sensitive or sensitive to this necrotrophic effector. And most of the sensitive lines were from South Asia, Ukraine, and Canada. We got similar histogram with PTR tox B. 31% of the Durum panel was sensitive to this necrotrophic effector. And most of the sensitive lines were from the Middle East, Southern and Eastern Europe, and Australia. The Manhattan plots we got with the PTR-tox and PTR-tox-B infiltrations, they showed what we were expecting. So the significant effect of TSN1B1 and really strong peak or high peak with TSC2. Here I show you the scale that we used for PTR-tox-B. In infiltrations, we use a scale from 0 to 3, and 2.5 and 3 would mean necrosis. But as I mentioned earlier, with PTR-tox-B is inducing necrosis. It doesn't go up to like the necrosis symptom. Everything, all the genotypes with score 1 and above that were considered being sensitive, and everything under 1 were insensitive, sorry. When we were scoring the Durum population with my colleague Gurminder Singh, we saw this symptom, the chlorosis extension toward the leaf tip. And we were wondering how to score these leaves because obviously this is a stronger reaction than the leaf in the middle. So we decided just to score the infiltration area as we would normally do, like using this scale. And make a new data set just for about the expansion of chlorosis symptom using simply just 1 and 0, depending on if we saw this symptom or not. And this was present in 64% of the tox-B sensitive lines. So here are the Manhattan plots I showed you earlier. The Manhattan plot we got with PTR-tox-B infiltration data, and then the Manhattan plot we got with DW5 inoculation data. And we can compare now these two Manhattan plots with this. But we got with the expansion of chlorosis data, so the 0 and 1 scores. And as you see, we detected TSC2 locus. And then this QTL showed up on the long arm of chromosome 5B that was present with the tox-B producer DW5. And we wanted to exclude the effect of this locus, TSC2. So the was on expansion of chlorosis data using only the tox-B sensitive lines. And only this QTL showed up on the long arm of chromosome 5B. And it was also interesting that the group that showed the expansion of chlorosis symptom had a significantly higher disease score with DW5 compared to those lines which didn't have the expansion of chlorosis symptom. So that indicated that this QTL on chromosome 5BL is a pathogen virulence factor. To summarize the results we got on the global Durum wheat panel in the 10-spot test, TSR7 is a major resistance factor in Durum wheat. TSC1 and TSC2 play significant roles in conferring susceptibility. And as it was demonstrated earlier, TSN1 is not a relevant factor in 10-spot development in Durum wheat. And we found a new QTL on chromosome 2AS that was associated with race4 isolate. We found a new trait that was expansion of chlorosis toward the leaf tip, and that increased disease severity. And the associated QTL was mapped on chromosome 5BL. And taking all this together, breeders are recommended to select for TSR7 and eliminate TSC1, TSC2, and TSN1B1. I say TSN1B1 too because tox-A, TSN1 tox-A interaction is still important in case of Parastagonospora nodorum, which is also producing tox-A. We found 14 durum lines that were resistant to 10 spot or 10 spot isolates and necrotrophic effectors. And now I will talk about genetic mapping of septoria nodorum blotch resistance in durum and spring wheat. On this histogram that we got with the 5 necrotrophic effector we used for infiltration, you can, in the spring wheat, you can see that 60–70% of the spring wheat lines were sensitive or moderately sensitive to SN-tox 267 and SN-tox 3. And about 58% of the spring wheat lines were insensitive to SN-tox 1, and 73% were insensitive to SN-tox 5. The ratio of SN-tox, say, insensitive and sensitive lines were close to equal in the spring wheat. And in durum wheat, on the histogram that we got with the 5 necrotrophic effectors, the same necrotrophic effectors that we used in spring wheat, you can see that the pattern is close to the opposite compared to the spring wheat. So the majority of the durum lines were insensitive to SN-tox 267 and SN-tox 3. It's about 80% of the population. And 75% of the durum lines were sensitive or moderately sensitive to SN-tox 1. About 2/3 of the population was sensitive or moderately sensitive to SN-tox 5. I mentioned at the 10 spot part of my presentation that about 1/3 of the population was sensitive to SN-tox A. And again, in the manuscript that I mentioned earlier, that was published in 2020, and the durum population from the National Small Grain Collection, they found about 62% of the durum lines sensitive to SN-tox A. So taking those results together with ours, we can probably say that the SN-tox A sensitivity is similar in spring wheat or hexaploid wheat and tetraploid wheat. And now I will show you the Manhattan plots we got with on the hardest spring wheat panel during the SNB resistance test. As you see, the TSN1, B1, SN-tox A interaction was significant in the panel. Just like the SNN1B1, SNN1B2 had a significant effect in the panel. And we found 2 loci associated with SN-tox 267 on the chromosome 2D. And these were SNN2 and SNN7 loci. We also found the QTL on the long arm of chromosome 3B. That was a new, that is a new locus associated with this necrotrophic effector. But we didn't identify the effect of SNN6. And that was not surprising because SNN6 was found only in a synthetic hexaploid line earlier. So we didn't really expect SNN6 here. And we found the significant effect of SNN3B1, SNN3B2 in the hardest spring wheat panel. And similarly, the SNN5B1, SN-tox 5 interaction was also significant. And in the global durum panel, the effect of SNN1B1, SN1B2 locus was significant with SN-tox 1. And with SN-tox 267, we found 2 loci associated with this trait on chromosome 2A. and another QTL on the short arm of chromosome 6A, but this is not SNN6. SNN6 would be on the long arm of 6A. So this is a new QTL here. A related study that was about cloning or fine mapping SNN2 and SNN7 in hexaploid wheat suggested that these two loci on chromosome 2A are homologous, might be homologous loci of SNN2 and SNN7 from hexaploid wheat. The effect of SNN3B1 and SNN3B2 was significant with SNTOX3. And interestingly, the effect of SNN5B1 was much lower here in durum wheat than in the spring wheat panel when we did the GWAS with the SNTOX5 infiltration data. And there was another QTL on the long arm of chromosome 7B, and then strongest effect had the locus on the short arm of chromosome 2B, and that was termed as SNN5B2. To take all this together, the hardest pink breeders, because of the high frequency of SNTOX-A, SNTOX267, and SNTOX3, they should eliminate TSN1B1, SNN2, SNN3B1, SNN3B2, and SNN7. And it is recommended to select the resistant allele at the 3BR QTL that was associated with SNTOX267. And we identified 41 lines insensitive to all 5 necrotrophic effectors in the hardest swing wheat panel. And the durum breeders, because of the high frequency of SNTOX-A, SNTOX1, and SNTOX5, they should eliminate TSN1B1, SNN1B1, and SNN1B2. And it's also recommended to select resistant alleles at the QTLs that were associated with SNTOX5, so the QTLs on chromosome 2BS, 4BL, and 7BL. We identified 32 lines insensitive to all 5 necrotrophic effectors. And it's important to mention here that we found 11 lines total that were insensitive and resistant to both SNB and 10-spot. The list of these lines have been published, and I will show you at the end of my presentation the manuscripts where you can find those. I'm not showing here in my presentation. And now I will show you some related studies where the phenotypic data of this research were used to validate markers. So Dashish and Aviratne identified 2 copies of SNN1 termed SNN1B1 and SNN1B2. She found that SNN1B2 evolved relatively recently compared to the paralog SNN1B1, and she developed Cas markers eliminating sensitive lines and the SNN1 null allele Cas marker. And she validated those markers in the global durum panel and the hardest spring wheat panel. This manuscript came out just a year ago, 2024, June. Zhangxi Zhang, she was working on SNN3. She published the manuscript in 2021, where she cloned SNN3D1 from Agulap's Tashai, and a candidate gene, SNN3B1, was identified that was recognizing SNTOX3. And later, while she was characterizing and validating SNN3B1, SNN3B1 was identified, which also recognizes SNTOX3 and leads to susceptibility. She developed markers for showing the absence of SNN3B1 and SNN3B2 and used the hardest spring wheat panel phenotypic data to validate those markers. And this manuscript came out earlier this year. Catherine Running identified two loci controlling tox-A sensitivity, termed TSN1B1 and TSN1B2. She developed breeder-friendly markers to identify tox-A insensitive lines and validated the markers in the global Durum panel and the hard-dressed pingwheat panel. This manuscript was published just a few days ago. Gurvinder Singh, he was working on the gene TSC2. He did the molecular cloning and characterization of that gene, analyzed the allelic diversity of TSC2, and developed diagnostic genetic marker for marker-assisted elimination of this gene, and validated the marker in the global Durum panel and hardest pingwheat panel. And this work is under the publication process. And at the end of my presentation, I'd like to thank to Dr. Stephen Shu, who let me work on the global Durum panel. It was a great panel to work on. I really enjoyed it. And I thank to Dr. Justin Ferris, who was my advisor during this research. And I thank for the researchers in his lab, Zhangzi Zhang, Amanda Peters Haugrud, Catherine Running, Gurminder Singh, Sudashi Senaviratna. They helped me in infiltrations, data analysis. Catherine helped me to find candidate genes in the QTA regions. And Gurminder, Sudashi, they helped me to score infiltrations. Gurminder helped me in the ToxP infiltration and Sudashi in the ToX1 infiltrations. Megan, Kaylee, Stephanie, Haven, they were technicians in the lab. And Dr. Timothy Friesen is a plant pathologist at the USDA ARS in Fargo. He and his technician, Daniel Holmes, they helped me in the SNB inoculations. And Dr. Zhao Huiliu from NDSU and his technician, Ganjin Shi, they helped me in 10 spot inoculations. And I'm grateful for my current team members. Dr. Elias Elias is the Durum wheat breeder at NDSU. Evan Salzman, Jason Axman, and Sally Mann, I appreciate their support. And I thank for Filippo Bessi, Marco Maccaferri, Roberto Tuberosa, and Luigi Cattivalli for their collaboration. We got the Global Durum panel from them and the genotypic data of the Global Durum panel. And my research was funded by the RISE program. And here are the two manuscripts I mentioned that was published. These were published on the research I showed in my presentation. In 2023, in phytopathology, we published the mapping results on 10-spot resistance in the global Durum panel. And the second one was published earlier this year in plant disease, and it was about mapping SNB in the Durum panel and the hard-dressed pingwheat panel. And thank you for your attention, and contact me on my email or LinkedIn if you have any questions. Thank you. We can't hear you, Josh. You muted.
All right, do people hear me?
Yep.
Oh, thank you for such a great presentation. Please, everybody, let's give Agnes some applause over the web. So far, we just have one question that I'll read to you. It says, based on your SN Tox 5 effector Manhattan plot, I realized you mentioned you found resistant QTL on 4BL. But from my knowledge, SNN5 gene is present on 4BL. Please correct me if I'm wrong. I think the QTL that you found on 4BL chromosome, it might be corresponding to susceptibility gene SNN5. Overall, your presentation was very insightful. Thank you.
So SNN5B1 is on the chromosome 4B. And And SNN5B2 is on the chromosome 2B. I don't know if I answered the question.
I guess was there in your SN Tox 5 Manhattan plot?
Yeah,
Did you find something on 4BL?
for B,
4B?
for B, for BL. Yeah, it should be on for BL.
Okay, so is it possible that that QTL is the same as SNN5?
Yes,
Oh, okay.
yes, it was marked as that, marked as SNN5B1.
Oh, okay.
Yeah.
All right. Here's a second question. How old uh how old are the plants when you do the inoculation with Pyrenophora
Uh,
candida?
I did this test like almost years ago, to be honest. But it's I think it's still 2 weeks, 2 weeks old or 10 days, something like that. Yeah. So yeah, the second leaf should be uh, big enough. So yeah, yeah, it's 10 or 14 days. I can't really remember, but it's in the methods in my manuscript. So
So you do the toxin infiltration in indoor grown plants at about 2 weeks.
yes, yes.
And do you find that this assay for toxin sensitivity correlates well with sensitivity of adult plants in the field or susceptibility or resistance? Do you see a
Well,
good correlation?
we didn't have field experiments in this research, so
Mhm.
I personally can't tell you that, but yeah, it should correspond. Yes.
Okay. All right. We have a question. Which software did you use in your study? I think you mentioned you were using
R
R.
R
Yeah.
yeah R
And
Gapit.
maximum likelihood, MLM. Okay. Here's a question. Which method did you use to express SN tox 267 and
260.
SN tox 5? Do you have any information about SN tox 4 infiltration? Thank you.
We
How
did
do you
not.
get how do you get the toxin, I guess?
The cultured filtrate were prepared in our lab, and then so we did the infiltration. Those were those culture filtrates, like I showed on the picture in my presentation, and we did not test Ascentox 4 because yeah, it hasn't been purified yet. So
Mhm. Okay, there's no more questions. I guess I had a question. I'm not all that familiar with all of the literature. You mentioned some of these toxin susceptibility factors have been cloned and identified in plants, and I'm curious as to whether the identities of these genes, the function of these genes, whether they are perhaps in a common pathway, or are they just sort of unrelated uh, genes?
the pathways can be different, but I think we are in the process of cloning genes faster and faster because of the new technologies. So I think we will characterize the new genes faster and
Mhm.
yep,
Yeah.
yep.
So how many have been actually cloned and characterized?
Well, I showed you on my presentation that most of them have been, but we find new loci and genes associated with sensitivity. So
Mhm.
yeah.
Okay. All right, here's some other questions. Should I do the experiment in the field to the yield as an end product after infection effect? So, um, so how do you, I guess this person's looking for advice on doing um, testing in the field for pathogen testing.
I think it's really hard to do these experiments out in the field because there are more things going on. So it's easier in a controlled environment and with the infiltrations or inoculations with isolates that we know. So we didn't have any field experiments.
Yeah. So the next question addresses, so mostly you talked about susceptibility genes, but I think you also mentioned a resistance gene. So if you have TSR, the resistance gene, and you have the susceptibility gene, TSN, together in the same host plant, what would be the disease reaction? Would it be resistant or sensitive?
uh, so what would be the combination
So
TS?
TSR and TSN. So I guess you have the
If both of
susceptibility.
them would result to resistant, or so the TSN is missing or
No,
is there?
present. So you have
Present. Okay,
both
so
a
okay.
susceptibility gene and a resistance gene. So
Oh yeah, I got it.
yeah.
Um, that's a good question. Um, uh, well, I guess it depends on the isolate that is going to infect the plant. You know, if it's producing uh, toxin or not. Um, yeah, it depends on the isolate that is going to
Hmm.
infect the plant.
Okay, and then this was actually a two-part question. So for the resistance gene TSR, has the AVR gene, the necrotrophic effector gene, been determined yet?
No,
No. Okay. All right.
no.
And here's a question: how did you characterize the pathogen race? So,
Um, yeah, there is a method. They characterized it or grouped them by what kind of symptoms they induce. So necrotrophic reaction or chlorosis, and depending on there are differential lines they call, and then depending on the reaction of the differential lines, they group them into these races. Yeah, but this might change in the future because it's getting a little bit too complex. It looks like it's not working as clear as they thought earlier. So there, as we saw in my presentation, there is difference between hexaploid and durum wheat reaction. So we can't really just group wheat together. Yeah.
okay, there's no more listed questions. I might just ask uh, one last question. Yeah, so you have the susceptibility genes, which produce factors that are sensitive to a toxin, and presumably you have alleles of these uh, receptors that don't interact. And I'm wondering to what extent that's been studied, sort of the allelic variation, and whether you can actually get at, what part of the protein would bind to the factor or whatever. Any more like in-depth molecular studies being done?
Yeah, it's actually this type of work were done in the manuscripts I showed at the end of my presentation, and yeah, there are different alleles. So they did research in the allelic diversity of these loci or sensitivity genes. So
Okay.
there is information in those.
Okay, sounds very interesting. All right, well, I want to thank Agnes again. Please, everybody, thank Agnes for a great talk. I want to thank all of the participants today who showed up, and we all appreciate you out there. And one more, there's one quick question that just came in. It says, in my understanding, AVR effector type resistance is based on hypersensitive reaction triggered by effector recognition. Does TSN1 varieties show HR reaction?
It's not about a virulence genes. These are necrotrophic effectors. In the necrotrophs, we talk about necrotrophic effectors. Um, so I'm not sure if I understand this question
I
at the
guess,
end.
well, would you say, is the TSN1, is that a classic R gene? Uh,
No,
no, okay.
no,
So it's
no.
all right. So we'll just leave it at that. Okay, once again, let's all thank Agnes for a great talk and thank all of you for your participation. And we will see you next time in October. Okay, so everybody enjoy your day. Thank you so much.
Thank you.